Hi.
Hello, how
Hello.
are you? How are you?
I'm pretty good.
Yeah.
I actually got my diagram to mostly work.
Oh, yeah?
Mostly, yeah.
Well, that's great. So it's
And
good.
nobody else is on yet.
I don't know. Yeah, I don't know if Dick's going to be here or not. There he is.
Oh, there he is. Oh, okay. Hi.
Hi, good morning.
Hello. Hi.
Hi.
I don't.
Okay, Bradley, you've got a problem.
Yeah.
No one showed up this morning at all.
Ah,
Okay.
yeah.
Now, I'm not sure exactly what to do about it.
Yeah, I know it's going to be hard to get people corralled at any one time. I don't know.
And you're not having much luck here either with anybody from India.
Yeah.
It's crunch time for students.
Yeah, it is. It's around,
I suppose
getting around.
in the case of our breast cancer group, they're not all students. So,
Yeah. No.
Oh, do they figure
Yeah.
it?
only 2 of them are students.
Yeah.
Do they figure it's Thanksgiving?
Well, they don't have Thanksgiving there.
No, they're not.
Oh no, but do they know that it's Thanksgiving and figure you're all on Thanksgiving?
Oh, I don't know. I'm in Canada.
Yeah, they're not. Well, yeah.
Hey, happy Thanksgiving.
Canadian
I already
today.
got it.
Yeah.
They Canadian Thanksgiving?
No, Thanksgiving was a month ago.
Yeah, I thought so. Okay,
Yes.
right. Yeah, I know the U.S. one is coming up in a week, but
Oh, yeah.
okay.
I don't know. Yeah, I don't know. I mean, I guess you can just keep sending emails to people, but I don't know if they respond. People just, yeah, it's going to be kind of tough to coordinate because people are busy doing things, and the people in Afghanistan, like when they have power.
When they have power, right?
Yeah. So I don't know. Another thing I found is like you can do asynchronous stuff, so you can go and have like do things with one group and then transfer to another, transfer that what you did to another group, like
Oh,
yeah, like just basically have a summary of what was done and that sort of thing. So it's not like they get the full experience, but at least they're in the loop as to what's going on. I don't know if that can be managed through a website or through like something like that, but it definitely helps people know what's going on.
okay.
Yeah.
Yeah, I have no experience with some of this. I just
Yeah.
Yeah, well, we have our expression, herding cats.
Yeah. Well, yeah.
yeah, yes. My daughter-in-law does that for a living, herding cats.
Oh, herding cats, huh?
Well, yeah, she has a bunch of group homes that she looks after, trying to keep everybody in line.
Oh, okay. I've done it once in my life. We had a whole bunch of cats, but they all ran downstairs simultaneously.
Oh, they did. That's
Yes.
amazing.
Yes, once.
Oh, once. Oh, okay.
Once in a lifetime experience. No.
Oh well.
Okay.
Okay.
Okay, I'm not sure. I'm thinking about how to draft a note, but
Yeah.
maybe I'll do it with you.
Yeah, that'd be good. I could help you.
Okay.
Yeah.
Okay, you got more experience than me in this.
Yeah.
Okay.
All right.
You
So
want to proceed?
yeah, yeah. So I've been doing some stuff with some of the things that we've been working on. I'll share my screen on this. So the first thing I have is this demo of embedding elements in noise. I'm not sure this is what we're thinking about, but I was working with Scilab, which is a program that's kind of like MATLAB, but it's open source, so you can work on matrices and transforming matrices. And I've been doing this noise embedding, but I've also been doing some of the stuff from the tensegrity book in Scilab and trying to play around with that, which actually has worked out. I've been able to get
What
that
is
code to run. The tensegrity book that you actually pointed out,
Oh,
yeah, they
now
had
what is
code in
this?
there for different, right? Like taking
Yes.
a matrix, describing a tensegrity network in terms of the forces, and then getting like a rank, and it's like doing matrix calculations and getting an output like a rank of different matrices, and then plotting that out.
Yeah.
And so you can put in any set of values for the tensegrity matrix, but it's usually, I mean, you can make comparisons between matrices. So if you say this matrix represents this structure, and these are the forces, and you get like something at the end that's like a rank of the matrix, and so it gives you a measure.
You've been doing that? You've been using it?
Yeah, I got it to work, and I mean, all I need to do is just like create a matrix of a certain size, and the values are like basically -1, 0, and 1, which represent certain aspects of the physics. So, like if there's a dynamic tension, it's like 1. If it's neutral, it's 0, and if it's not, it's -1. And so it's like at each node, there's a certain property to that node if it's in dynamic tension or not. I can't remember the criterion, but it's pretty easy to follow through. You just put in a matrix, and you can get a measure at the end, and you can do that for a
Okay.
number of matrices. So you could actually design your matrices saying that this represents this thing, and then so that works out. I was able to get that to work.
Okay. In what language?
I can just use it in this Scilab program, which is basically like MATLAB.
Okay.
It's not well. I mean, it does most of the things. It does everything MATLAB does, except for some of the simulation stuff. But but the book from the code from the book works. So that's it.
Oh,
Okay, Bradley, I don't understand the expression. What are you embedding in noise?
So let me go through this. Yeah. So this is a Poisson distribution. This is actually just defining a Poisson distribution in terms of its gamma value, which is kind of like its descriptive parameter. And the higher the descriptive parameter, the more points you get going, like kind of up. So like the lower the gamma, it's kind of resembles a Gaussian distribution. The higher the gamma, you get more values at higher. You get more higher values, more points, and so you get this type of Poisson distributed signal here. So that's what I have plotted out here over 10,000 points. So these are just points that are generated by this function. This is a random generator function for Poisson distributions. It's over 10,000 points. So this is the Poisson signal that's out in the world. And then this is where there's some noise, a uniform noise that's generated, these green dots. And so these are generated, and I can plot them over the Poisson to hide those points. So the blue points are my Poisson points that I generated in this step. And then here I've generated some noise overlaying these Poisson points. And so it's hiding some of the outlier points here, the higher ones of higher value, and the ones that are in the meat of the distribution are kind of still visible. But of course I can stretch this out, and to the eye, it appears to be more embedded in here.
What's in the blue line? It looks like characters. But what is
Oh no, they're just dots. So they're just points that are sampled. These are circles.
Can you expand that so we can see it close up?
Yeah, let me see if I can get in. No, it's not doing that. Okay. If I go backwards, it actually zooms in. All right. That's as close as I can get it, but so they're just independent points. It's just that there's so many of them.
I see.
Yeah, it just kind of overlap. So you have this blue line.
They're little circles.
Yeah, little circles.
That's why they look like characters.
Yeah. But yeah, there are a lot of them there. So, and then so they're just circles, and then these are also circles. These green, but because there's so many of them, you get this field of it's like a green field over these blue circle lines, and then so it embeds it. And I was like playing around with the aspect ratio of the image, and so you can get like more crowding. This is exactly the same code that generates both of these, but this strip here is narrower, and it apparently it embeds it more, at least to the eye.
Why is it different values at the top from the middle and the bottom?
You mean in this distribution?
Yeah.
Yeah.
Poisson distribution was. What's the sequence? In other words, going down the page.
So it's just that it goes from left to right. So it's generating things in time. And the idea is that it's like they call them arrivals sometimes in Poisson distributions, but you're measuring like a calculation of how many things you observe at a certain time. So it's like if you think about like a highway where you're standing on an overpass and you're looking at how many cars pass in like a minute. And maybe sometimes there are no cars or very few cars, and other times there are like 20 cars.
I understand that,
Yeah.
but I don't understand why all of the ones with no cars are up at the top and the ones with lots at the bottom.
The ones that the high values are up at the top. So these are the ones where you might observe 20 instances in one observation.
You mean there's an axis?
Yeah. So this axis is I cut off the axes because I wanted to have it as like a window where I just had the points.
Okay.
But I didn't put the axes.
The axis would help.
Yeah. Well. Yeah. For what? Yeah. Now that I'm explaining it.
So the time sequence is left to right. It's not a top to bottom.
Right. Left to right is the time sequence.
Okay. And what are the rows?
The rows are frequency. So it's like at the bottom you have. Yeah. So your mean, as it were, is about 4, I think, or 5,
Okay.
And then as you go up, you get fewer observations, up to like 16 in this case.
Yeah. Now I see what you're plotting here. Okay.
Yeah.
It wasn't obvious.
Yeah. And then
So the time sequence is strictly left to right.
Yes. And this is all just kind of like generating the data so we can create these masks, but that's how it's generated. So it's and then the uniform is just basically where it's generating random values but uniformly over the space. So you can see that there's this uniform distribution of points, and then that's overlaying the Poisson distribution. So you're hiding some points that are less frequent in the Poisson distribution, but the more frequent points that occur you can see through. And so the point of this then is to have like this mask over the distribution with a uniform mask that kind of hides some of those points. And then that's what I guess we wanted for detecting points out of the background.
Okay. Now suppose that we momentarily switched to a different distribution.
Yeah.
Okay.
Yeah.
Does that show up on this graph?
I could make one like that. I mean, it would you could generate it the same way and it would look a little bit different. But yeah, you could do that.
Okay. Why are you doing this?
Well, we wanted to create a mask. We wanted to create like signals embedded in some sort of noise or mask. So this is the uniform distribution that would be like the noise mask, and then the Poisson distribution is underlying that.
Okay, so you got me thinking in terms of detecting tumors.
Right.
Okay.
Yeah.
if in the time scale, your time scale left to right is kind of like could be a spatial coordinate.
Could be what?
It's sort of a spatial coordinate. In other words, for a given amount of time, you switch to a different distribution.
Yeah.
And the question is, if you did that, would it show up on
here? Yeah, it would.
Okay. And the extreme cases, for a given amount of time, the signal is constant.
Okay, yeah.
Okay, it would show up here, I suppose, as sort of like an elongated ellipse or something like that.
Yeah. Yeah. So this is, this is just the code here that is run in Scilab. So this grand function will model a lot of different distributions. I mean, you have to have the correct parameters, but it'll allow you to generate them, and then you can plot them one on top of the other. Yeah.
Do they have that Perlin distribution?
They did. I was looking into Perlin noise. They don't have it in the grand function. It's a little bit more complex, but I think it might work. I'm working on it in Scilab. It would be nice if it did, because you could actually run it. It's a procedural thing, so it would require like building a for loop and getting things. Yeah.
Can you write macros in this?
I think so.
You think so?
Yeah.
Okay, so maybe you can maybe you can expand the language.
Yeah.
Okay.
And so then this might actually answer, I don't know if it answers it, but you can transform this Poisson distribution in different ways. So in this case, I've permutated the signal, and I've done a transform of it. And this is where this distribution that you see here, which is a Poisson distribution, that's been sort of permutated and transformed so that most of the signal is down at the bottom. So it's from that distribution, but it's been permutated. And so now I have a band at the bottom where you can see it's a little bit more visible, and you can do things like that, or you can do another one where there's a band at the top. It's just generated from that same distribution, but it's transformed somehow. So, yeah. And so that's my demo for that. I don't know. I mean, I know it's probably food for thought.
Okay. Yeah, it's taking a while to swallow.
Yeah. So then we have this Ensembles of Early Life paper, and that's something that Dick and I are working on with someone else. And I've been working on the Braitenberg Vehicles section. We've gone over it a little bit, and I'm going to go over the paper on, I've been working on the Braitenberg Vehicle section, kind of I don't have that version on this open here, but I'm working on some of the details in terms of how this might work, how we might be able to, like the parts list and how to make that sort of more salient, because I know I sent you a section, and then you made a table, and you said, here's a parts list that we might want. And I've been working on that, refining it.
Oh, okay, good.
Yeah.
Good. Okay. Yeah, that'll be interesting to see how many random structures actually function.
Yeah. Yeah.
Okay.
Yeah.
I'm not sure what the criterion is, because with living organisms, let's say microorganisms, you can get weird mutants that go around in circles or make patterns or do all sorts of things other than swim normally.
Right.
And I guess you'd still call them alive.
Yeah.
It's just that they have a different function in life.
Yeah.
Okay.
Yeah. I know that.
Like we heard, there's a crazy guy who's he calls himself the magnetic reversal group. But magnetic reversal, I don't know what you call it. It's not a podcast because you can see him.
Yeah.
But he was reporting on a bunch of sheep that go around in circles all the time.
Oh, yeah.
Okay. Now, he's attributing this to changes in the magnetic field, whereas it's a well-known disease in sheep.
Oh.
Okay. The sheep just keep going around in circles. So, the sheep are alive, but they have a different motility from normal ones.
Huh. Yeah.
Okay. So, What? Do your cows go around in circles on you?
No,
No.
no, the behavior that I know about is if I sing to them, they run the other way. That's fast.
Yeah, well, okay. If you sing to a piece of meat, it doesn't run. So.
As a follow-up to this stuff on early life that we were talking about, I'd like to go over a paper on autocatalytic sets. So this is a paper from Stuart Kaufman, Wim Hordijk, and Mike Steele. These are some pretty big names in complexity theory and modeling. And this paper is called Autocatalytic Sets Arising in a Combinatorial Model of Chemical Evolution. So this ties to the origin of life. This is talking about chemical evolution in the origin of life. So this basic idea is not new, but it still leaves open questions about how exactly it could have led to a coherent self-reproducing collective of molecules. So the idea is that you have chemicals in the environment of an early planet, in this case early Earth. How did they come together to build coherent and self-reproducing collectives? And so that's the question they're answering here. One possible answer was proposed in the form of emergence of an autocatalytic set, a collection of molecules that mutually catalyze each other's formation that is self-sustaining and gives some basic food source. So you need a couple of things. You need one is that mutual catalyzation, because you can't have a master catalyzer when a system is self-organizing, and something that's self-sustaining through some sort of food source. So there's got to be some sort of metabolic input. Building on previous work, here we investigate in more detail when and how autocatalytic sets can arise in a simple model of chemical evolution. So they're doing this modeling exercise to show how these might arise. This is based on the idea of combinatorial innovation with random catalysis assignments. So this is just kind of an innovation model that they use. We derive theoretical results and compare them with computer simulations. So this is a lot of these early life experiments using chemical evolution were done by O'Perrin and Haldane. Of course, these two people are very important in the early, in the origin of life and in evolutionary biology. So that they've, these are big names in those areas. It is later shown to be plausible through experiments of Miller. So this is from Miller-Urey, the Miller-Urey experiments. Recently, different variants of these experiments were repeated and analyzed with state-of-the-art molecular analysis technology. So I think number 4 is, all right, there we go. So the citation number 4 is chemical analysis of a Miller-type complex prebiotic broth, part 1. And then 5 is this part 2, gas, oil, and water, and the oil-water interface. So years ago, they did these experiments. These are the Miller-Urey experiments, production of amino acids under possible primitive Earth conditions, where they took a broth of different molecules, they put an electric charge across them, and they produced a lot of different metabolites. And they redid the experiments more recently, back in 2015, actually a little bit earlier than that, and they found that they got a much richer array of biomolecules to result from it. So the original Miller-Urey experiments, that was kind of a proof of concept, and then they redid the experiment later, and they found that it was actually even richer in terms of what they could get. So, some of these things are, maybe underexplored. So it's just something to think about. We're thinking about early life. But this paper actually goes through a number of different computational models or theoretical models for understanding this. It is an experimental. The first method is the TAP model. The TAP model is based on the idea of combinatorial innovation, which is where you have a number of different combinations that can be formed, and this leads to innovation in certain ways. So if you recombine things over and over, you get innovations that emerge. And so they use this equation here, where m sub t is the number of different things at time t, and alpha sub I is the decreasing sequence of probabilities. So this is again with this stuff where we talk about generating a bunch of configurations at different probabilities. This combinatorial model, the TAP model, actually probably would do well to describe what we're trying to do with the Braitenberg vehicles. And so, interpreted in the context of chemical evolution, new molecular species are produced through chemical reactions and arbitrary combinations of already existing species as reactants. So you have a number of species, chemical species, that is, as reactants, and you recombine them and you get these new combinations, some of which are going to be viable. And so this is exactly the kind of thing that happened in the Miller-Urey experiment, except it happened in great parallelism, and you have basically a huge broth of possibilities there. Note this above equation, this m sub t plus 1, and solving for that represents a deterministic version that does not guarantee m sub t to be an integer value and only serves to convey the general idea behind the model. So these are stochastic implementation for their simulations here. For each newly created molecular species X and each of the existing molecule types Y that were not present at time 0, X can catalyze the formation of Y with a fixed probability P. Similarly, for each chemical reaction R that produces a new species, and each of the existing molecule types Y that were not present at time 0, Y can catalyze R, also with probability P. These random catalysis assignments are assumed to be independent across all pairs of molecules and reactions. So they go through and they do this, that there's an algorithm, TAP with catalysis. They give the algorithm. This is pseudocode. It basically describes this process. Then over time, the m sub t existing molecular species and the particular chemical reactions that produce them thus form a growing chemical reaction network, which is shown here. There's a food set consisting of the m sub initial species as an example here. So this is where you have random catalysis assignments in the dashed arrows, and this feed-forward network with the solid arrows. So this is over time. This is over m sub 0, m sub 1, m sub 2, m sub 3. You end up at 10. So that's what their network looks like. They also have RAF, which is now the probability that such a growing chemical reaction network at some point a subset of molecule types exist in which the molecule mutually catalyze each other's production, which is sustainable on the given food set. So RAF then is this idea of reflexively autocatalytic and food-generated set, RAF. More formally, RAF is a set of R chemical reactions and the molecule types involved in them such that, number one, each reaction in R is catalyzed by at least one of the molecules involved in R. And two, each molecule type involved in R can be created from the food set through a sequence of reactions from R itself. So then they use some sort of algorithm to find such RAF sets in an arbitrary chemical reaction network, or determine that no such subset is present. So either RAF is present or not present in some arbitrary reaction network. This algorithm finds the unique maximal RAF, which is the maximal set of reflexive autocatalytic and food-generated molecules, and is the union of all possible RAFs within a given network. So then TAP and RAF can be used in tandem. You can look at RAF sets in the TAP model, and this was done in the context of technological evolution in 21, which is this paper here, Dynamics of a Birth-Death Process Based on Combinatorial Innovation. And this is where you're looking at the birth and death of combinatorial innovations. So you're looking at innovations that die and are born, and the process there. So you're getting, you could apply this model to other things other than the origin of life. But in this case, they apply it to the origin of life. and they do a lot of modeling here for different parameter values and show that this model is fairly effective at doing what they want to do. And I think there was some A-Life work where they did like these little morphological robots where they have the like full 3D, yeah, and they would deform them in some ways and see how they'd swim or run. And,
right.
Yeah.
It's been 20 years since I've seen those. But
Oh
okay.
yeah.
Yeah.
Yeah, I think people just kind of made them as a proof of principle, but I don't think they have like a genome or anything. They're just kind of like they evolve, I think, the controller to locomote instead of like the actual morphology.
Yeah, they're
Yeah,
sort of stick animals.
yeah, stick animals.
Yeah, and you make things move, and yeah, right. Okay.
Yeah.
Yeah, it's been a while. Yeah, that literature probably we should cite that literature.
Yeah. Making a note here. All right. Yeah. So that's great. So that's where we are on that too.
Okay, great. Okay. Any idea on that criterion as to whether a random structure is a vehicle?
Yeah, we're working on a couple of criterion.
I guess if there's no wheels, it doesn't move.
Yeah. Or if there's one wheel, I mean, it's really about like figuring out, what the structure, what function is, because function
Yeah.
can be, kind of surprising if you're just putting
Yes.
parts together.
Okay, right. I guess if there's a way of doing what the artificial life people did, and that is you put it together at random, and then you see how it moves.
Yeah.
Okay.
Yeah.
And then you could say, okay, if it can't move from a spot, like if it goes around in circles at one point, then obviously it's not viable in the sense that it couldn't get any food.
Right.
Okay, that would be an example. So the distance it can move per unit time might be a criterion for how much its function is working.
Right.
Oh well.
Yeah. Yeah. Well, yeah. So work in progress on that.
You're dragging me into psychology.
Well, actually, I think a lot of that was robotics because they're interested in like how a robot might move if you add or subtract parts. If maybe there's something that's very optimal if you have a weird morphology. Like.
Oh yeah. You're familiar with the tensegrity robots?
Yeah, I am. I know they've done a lot of work with that.
Yeah. So that's a possibility, and I know there's some sort of self-repairing, and their robustness is supposedly good.
Yeah.
There was a program that looked at tensegrities, and they kept taking out the struts that weren't necessary because they weren't being used or didn't have any pressures on them.
Okay.
And it started off kind of square, or what you'd call pointy, and then it gradually rounded, became a more rounded structure. It's all that paper. So
Yes.
very interesting.
That's not.
Oops, sorry.
Yeah, I don't think I've run across that paper, but I've heard of the tensegrity robots.
That's not our phone.
Yeah, Susan. Yeah.
Okay.
Yeah, that's great.
I'm trying to think there's an analogy. So I've been looking at a minimal cell.
Right.
And the way they make a minimal cell is they take a very small cell and they keep removing stuff.
Right.
Maybe by mutations or whatnot. And they got them down to only 70,000 parts. But
Yeah.
Wow.
the thing is, what got me going on this is a paper where they created a minimal, what they call a minimal cell, and then they did a simulation of it. And the simulation seems to imitate.
Yeah.
Right? And the remarkable thing of this is that, which they didn't emphasize at all, is that the contents of the cell are what counts, not the spatial organization. Because in a sense, the spatial organization takes care of itself because the fusion is very fast.
Right.
Okay? So I think they got this thing both metabolizing and cell dividing, and both in the living cell and in this computer simulation of it.
Okay. Yeah. Actually, I think I have that paper in the queue here. I was going to talk about it
Oh,
today.
okay. That would be nice to see.
Yeah.
Yeah, I really need you to look at that paper carefully because I want to make sure I'm not over-interpreting it, especially because they did not draw these conclusions.
Right. Right.
Okay? It's sort of like somebody discovers something and has no idea what they found.
Yeah. Yeah. What do you compare it to? Yeah.
Oh, it's a piece of mud. No, it's a diamond covered with mud.
Oh,
So I think this is what you're talking about. This is actually, I think you sent me this copy, but I saw a talk on
Yeah.
this at a conference this summer. This person, Zan Luthi Schulten, is at the University of Illinois, and they
Yeah,
do a
that
lot of
was
stuff with and they did this at a conference this summer. They talked about it, and they're doing this stuff with this is fundamental behaviors emerge from simulations of a living minimal cell. So this is where you're building a minimal cell. You're taking out like everything that isn't required for the cell to function, and then trying to build a cell that has like the bare components. So oftentimes like in bacterial cells, they'll take out all the genes that aren't essential, and they'll have this list of essential genes. And you might want to do that, say, to like if you want to build a bacterial cell that can just function metabolically, and then boot it up with genes to do something like, some task that they want the cell to do. So that's why you might want a minimal cell. It's just the basic metabolic requirements of a cell without any other function, really. So this is a graphical abstract. This is where they actually, I think they're working with yeast cells, but I think they also are doing some work with bacterial cells, and they're doing the cryo on it.
I think it was a reduced mycoplasma.
Oh, mycoplasma, okay.
Yeah, mycoplasma used to be touted as the smallest living cells.
Yeah,
I think that's what they started with. The mycoplasma, I guess, has been classified as bacterium.
Yeah, as a circular genome, I guess.
Maybe, I don't know.
Yeah, but yeah, so they do the initial cell state. They characterize the genomics, proteomics, and metabolomics. I mean, they have some money here for this, and they do basically characterize a minimal cell. Then they have these, yeah.
And they said I think they said they've been at it 10 years.
Yeah. It's a long time to get like one project.
Yes. It's a non-trivial thing.
Yeah. Then they have these receptors here and ribosomes. So they're looking at kinetic data from experiments. They're looking at cellular processes. Then when they get that, like they have the biological side, then they build these whole cell simulations, which are these dynamical systems that predict the cell, what the cell does. And then they compare it with the biology, so you get a good comparison of a simulation with the biology. So their simulations are highly detailed and highly, like they want to align the data from the biology of the simulation. So they do this they run it on a GPU, the simulation, and it's, they're able to extract different parameters, so these time-dependent cell states, looking at mRNA, cell doubling time, and ATP cost. So this is a 3D spatial resolution of a fully dynamical host cell kinetic model. Then you have these single reactions that are pretty detailed. You have these time-dependent cell state measurements, which are, of course, this function here. They talk about mRNA half-lives and looking at like a process where, if you have a transcriptional process, you can look at the mRNA half-life of that process. So if something is transcribed, how long does the mRNA live in the cell? And I've done experiments like this where you can shut off the mechanism and see what the mRNA half-life is. And it tells you something about the underlying mechanisms of metabolism. And then connections among metabolism, genetic information, and cell growth are revealed. So they actually can do a lot of these. It's not just like looking at a minimal genome, it's looking at the whole cell and how it's behaving. Let's see. So they have this whole cell fully dynamical kinetic model, which they call WCM, whole cell model of JCVI Syn3A, which is this minimal cell. And that's like again, it has a reduced genome of 493 genes. So Dick made a note on that has retained few regulatory proteins or small RNAs. So this is where they're not interested in like the functional regulation. They're interested in just kind of like the essential metabolic genes. They're doing cryo-electron tomographs to get sort of the image of the cell. So they're imaging the cell so that they can put it into a computational model of the cell geometry and ribosome distributions. Time-dependent behaviors of concentrations and reaction fluxes from stochastic deterministic simulations. So they're doing different types of concentration and reaction flux simulations that involves a lot of flux balance analysis and all that fun stuff. So that's not trivial. It requires a lot of computational power, but that's part of the simulation as well. This reveals how the cell balances demands. The cell balances demands of its metabolism, genetic information processes, and growth. So there's this, they want to see how these things are, basically what the cell is investing in each of these things, and offers insights into the principles of life for this minimal cell. The energy economy of each process, including active transport of amino acids, nucleosides, and ions, is analyzed. WCM reveals how emergent imbalances lead to slowdowns in the rate of transcription and translation. So that's interesting that there are these imbalances in some of these things, like in the metabolism, that actually changes the transcription and translation rate. So again, like when you have transcription, you're measuring it from the production of mRNAs, and you can watch the sort of the concentration of those. And in terms of slowdowns, the mRNAs have a certain half-life, so they survive in the cell, and if the number of mRNAs falls, the concentration of them falls, and you can assume that function has been shut down or diminished in some way. And if it's, if there's a higher concentration of mRNAs, you assume that process is enhanced. So you can do a lot of different things just by looking at things like mRNA half-life, what they call. So mRNAs, again, they don't stay around forever. They kind of degrade, and then you can actually look at those profiles and see what the kinetics of the cell are. Integration of experimental data is critical in building a kinetic model from which emerges a genome-wide distribution of mRNA half-lives, multiple DNA replication events that can be compared to qPCR results So these are where you can actually do separate validations using quantitative PCR and then experimentally observe doubling behavior. So these are all things that you're just looking in the experimental system, and they're using mycoplasmas in this case, in this paper. And so this is a very simple cell, and it's very easy to manipulate and to observe. So that's why they use mycoplasma. And so we have been able to produce living cells with fewer genes than any known naturally occurring cell. So this is some of their work. So they've been at this 10 years. They published a lot of papers on different aspects of this. Like I said, I think they've done some work with yeasts as well in sort of validating a lot of this stuff. So this is a pretty extensive project. Yeah, a lot of this involves not just looking at certain measures, but doing a lot of flux balance analysis, which is really kind of a difficult thing to get your head around how it works, because there are these really huge networks that have a lot of feedbacks. Let's see. This JCVI SYN3A is a genetically minimal bacterial cell consisting of only 493 genes on a single 543 kilobase pair circular chromosome. So that's a pretty small chromosome, compared to the human genome or even any animal genome. It's circular and it's pretty much optimized. There's not a lot of there's very little regulatory or extraneous DNA in that circle, and they have 452 genes coding for proteins. So this is other, and then so let's see. This the SYN3A genome was synthesized based on the known genome sequence of the natural parent gram-positive bacteria Mycoplasma mycoides subspecies Capri. It's a certain strain here, GM12, and has been synthetically reduced to achieve a minimal genome, producing living cells that grow and divide in about 100 minutes, and have consistent spherical morphologies with 400 to 500 nanometer diameters. So this is the size of the cell. Their kinetics are such that they divide in about 100 minutes, and then they have these spherical morphologies, which are actually pretty small cells. So that's the kind of thing we're dealing with here. In addition to biochemical reactions, whole cell 3D spatial models require cellular architecture, including spatial distributions of ribosomes, configurations of the circular chromosome. So inside the cell, you have the spatial model. It's a three-dimensional model, and there are all sorts of things going on in the cell. And the idea is that, is the spatial distribution important, or is there a role, like Dick said, for diffusion? And the rate of diffusion, as it increases, makes the spatial distribution less important. The cellular architectures are reconstructed at the single cell level directly from cryo-electron tomographs, which are these images that they, because it's such a small cell diameter, they have to use something really high resolution to get good images so that they can actually kind of replicate the inside of the cell in some of these things. So let's see. If we go here to this part here, I don't know where we want to start. We consider here two models of the stochastic simulations. And this is in figure S3, which is down at the bottom. A chemical master equation description that assumes the whole cell is well stirred. So this is where they're making assumptions about the cell. So they're trying to do this kinetic modeling, this flux balance analysis. They're using different setups for it. So they use a chemical master equation and they work from that. And so a lot of times you will assume that the cell is well stirred, and it includes diffusion and rates of transcription, and so it has all these different rates involved in it, but it doesn't require any sort of spatial segregation of mechanisms. It's all everywhere in the cell has an equal chance of these parameters being correct. And so that's why they say well stirred. And then a reaction diffusion master equation description that requires macromolecules to diffuse to each other for reactions that take place in the spatially heterogeneous environment of the cell. So they assume that these rates are constant, but that they're these these macromolecules that diffuse, and they interact in different parts of the cell. So in our simulations, we record time-dependent particle counts of each molecule and intermediate fluxes of all metabolic reactions. And then they have a spatial model, which is where the position of each macromolecule within the cell is accounted for. We present results of the well-stirred model over a complete cell cycle. Unhealthy cells within the simulation ran out of phosphenol pyruvate, halting glycolysis. So this means that the unhealthy cells were not able to continue. For the spatial model, was that?
Yeah, that's what, see, there's an interesting parameter. So I got 84% of the simulated cells persist in the sense of, at least for the number of cycles they went, for those would still be classified as alive.
Yeah.
Okay, so only 16% were terminal.
Yeah.
Okay, so I was thinking if you took their model, not that I don't know if we can run their model. Is their model? Oh, stupid question. Is their model publicly available?
I don't think so. I think this is something that they run. I mean, it would be hard to run. You'd have to have like access to a pretty high-end GPU, I think, to run it. But yeah.
Okay, because what you could do is start with their model, which has an 84% alive rate, and then take parts out until it goes down to 0.
Yeah.
And that would tell you, in a sense, that the minimal cell at the origin of life might have to be.
Right.
Okay, because it has to have some viability, just, a little bit greater than 0%.
Right. Yeah.
And if it's greater than 0%, then if there's a mutation mechanism, it can then enter evolution and start getting better and better.
Right. Yeah.
Okay, so what I'm saying is there is a set of experiments to be done here with the simulation by removing parts.
Right.
And they did not seem to consider that.
Yeah. Looks like in part the unhealthy cells basically run out of certain metabolites. So I mean, I don't know if that's because it's not being produced or because it's I mean, I guess it's just not being produced at the right concentrations. So that, that's like what, I guess there's a mechanism that's there that in some of the healthy cells that isn't there maybe in unhealthy cells. So maybe that got removed somehow.
Yeah, it's analogous to the tensegrity structures where you remove struts that are not being used.
Yeah.
Oh, well. Oh, yes, and we heard from Stephen Levin.
Oh, okay.
Yes.
Yeah. He says he's in his 90s now.
Oh.
Oh boy. Okay, yep.
Yeah, so.
Yeah, I spent a week with him, I'm trying to remember when. It was a while ago. I think it was within 10 years or something.
Yeah.
But what we could use from him is a clear statement of what is the difference between his approach and Ingber's, and why he's so down on Ingber.
He says it's partly minimal energy. You have to look at minimal energy to keep your structure stable.
See,
And
for me,
requires a twist, which
What?
is interesting. It requires a twist. And I said that
Yeah.
to you already.
My complaint about English stuff is that the tensegrity structures that he simulates never change topology.
Oh, well, I'm assuming that mine are going to change topology, and I'm just looking at a sort of a very small window of time, and then I'll make different arrangements and take a look at that.
Yeah, I mean, for example, he had one computer experiment where they kept adding a weight, and the tensegrity structure acted just like a spring. But a living organism tensegrity structure would probably change structure on being put under tension.
Yes, and Stephen Levin says that even the struts and the strings of your model are also a tensegrity structure, well, which is what they are. If you look at actin, it's a tensegrity structure.
Oh, okay. Okay.
So you have to put that in there as well.
In other words, it's a fractal tensegrity structure.
Yes, exactly. It's exactly what he says.
Okay.
Right.
Okay. So, yeah, the problem is to get beyond Ingber, but be careful what you take from Levin.
Yeah.
Well, I can't do fractal structures off in forever. So I could try it for 2 levels maybe, and then sort of go, okay, fine, enough.
Yeah.
Yeah. I think also it had different nonlinearities in the elastic components.
Well, that's if it's a tensegrity structure, it will be non-linear elastic.
Oh, okay.
Just naturally.
Yeah.
Yeah. So I think I should try to produce that for the struts and strings of my model, and then just, like I say, call it a day.
Okay.
And see the difference between linear and not and tensegrity structures, and then say whether my measurements with this are going to be better than the just the continuum model. And that's all I have to do is just say, does this model here work better than the continuum model? And which one's more computationally intensive?
Yeah.
But that's it for my project.
Okay. Bradley, getting back to that paper then.
All right.
The questions I've got are to what extent does the spatial model, which runs much slower, what extent does it imitate the behavior of the well-mixed model?
Okay.
Okay, that wasn't obvious to me.
Let's see.
In other words, are they essentially the same or not?
Yeah.
Even though one runs much slower than the other.
Right.
But are the results the same?
Let's see if they have any figures that explain that. I don't know.
You know what mixes things in a cell?
What?
Convection.
Right.
No.
Well.
Yeah.
Not at 0.4 microns.
No.
Yeah.
Really?
Yeah, it'll do Marangoni convection, like where it's mixing locally.
I don't know. That's a good. Well, these cells don't have any actin, do they?
I don't think so. I don't really know.
They don't have actin.
No.
Well, they're non-motile. They
Yeah.
just sit and drink.
I think
if there's no force generator inside the cell, I don't see anything except diffusion being important.
Yeah. Yeah, I mean, I think it's where the macromolecules interact. If they're interacting in certain places, or if they're just kind of, you just assume that it's
they could zip
anything is
together,
happening.
something like that.
Yeah.
Convection is always present, even at that small size.
You sure?
Well, I'm pretty sure, yeah.
Okay,
He
well,
said reactions in microgravity have a different timing. They automatically have a different time sequence. So that's a molecular level thing.
yeah, but at what size cell? These are extremely small.
Yeah.
Well, it's.
These are points.
I would like to see how important it is at that size. It'd be interesting.
Yeah.
Okay.
It's not clear. And
okay, I understood what you're saying. So
yeah.
Bradley, can you? They probably don't mention convection, do they?
I don't think so. They just mentioned that things are either well mixed or not, and
Yeah.
a lot of this might be because you have, hydrophobicity of DNA, and at that scale, it's like that's really kind of because it's pretty packed in, as you see from the C.
Yeah.
So this is the cell. This is the model of the cell here. So we have this digital model of the cell. This is the image, or we're taking these coordinates from the image, and then we're building this model, and then this is what we're simulating. So you get like these macromolecules that are maybe clustered or not. They're moving around, and they can interact. They can do some reaction anywhere in the cell, which is of course very small. And then they do the spatial simulations. They do. I don't know if they really break it out. I don't know if they're comparing the two, well mixed and spatial. That might be further down. This is an example of the metabolic network here, so you can see how this is pretty complex.
The spatial simulation means that they give a coordinate to individual molecules, but they still move by diffusion.
I think everything is a coordinate. It's just that like the spatial model assumes that there's like I guess maybe compartmentalization almost.
Well, I think just that they know where everything is.
Yeah, well, it could be too.
But
let's see.
I assume it includes diffusion.
Yeah.
And okay, now the curious thing is I looked up the rates of diffusion in crowded cells.
Okay.
And diffusion rates seem for macromolecules to go down only by a factor of 2 or 3. So they may slow things down a little bit, but I suspect that crowding behavior does not make much of a difference.
That's interesting. So let's see if there's anything on these comparisons. This is transcription translation. They have some supplemental figures, which I'll get to in a minute here. They give spatial, well-stirred and spatially resolved. So this is a comparison of gene expression between well-stirred and spatially resolved simulations. So I guess the spatially resolved is where they have the coordinate information so that they know where, and then, so they just have accounts for RNA, mRNA for genes coding for genetic information, processing proteins over time. This is the mRNA count. This is the time. So you can see that there's in the spatially resolved cells.
Oh, okay.
Yeah, you have more sort of it fluctuates more, I guess, over time. That could just be an artifact of having the spatial information there, as opposed to the well stirred where you have just a count, basically. So let's see. Spatial model with lower concentrations of nucleotides than the well stirred model, which could be tied to 2 factors. First, more nucleotides are being incorporated in mRNA in the well-stirred model. And then second, the current spatial model does not include DNA replication, which initiates on average around 10 minutes in the well-stirred model. So this is, it still doesn't really give a lot of insight into that difference.
So on the parameters, they could measure the spatially resolved is about the same as well stirred.
Yeah.
Okay.
Let me see. Move down to the other supplemental materials, which are down at the bottom. I think. It's hard to know. Sometimes they don't. Like, you can see how much supplemental material there is here.
Yes.
Yeah. Actually, I don't know if they have them in this, so maybe not. Maybe I have to go offline and get them. But yeah, so this is, yeah, we won't go to the supplemental materials because I don't have them, but, or the figures. But I think that's, I don't know if that would give us any more insight into that. Let's see. Yeah, I think that's all there is for the different figures. I think this is the last one where they kind of give an example of well stirred versus spatially resolved. And they give these different conditions. So this is the mRNA count here. This second set is the concentration of intracellular pools of NDP, NTP, and ADP. So these are macromolecules. And you can see that there's a difference here. The spatial resolved one, again, has more variation and fluctuation. The scaled protein counts, these are number of unique proteins. There's a much difference there. And then the number of unique mRNAs are the counts for the half-life in minutes. So this is just things that are getting synthesized and degrading. So you get more unique mRNAs in the spatial resolved model, but it decays a little bit faster, at least according to this histogram. And let's see. So in terms of mRNA half-life, they're compared between the two methods, where the well-stirred half-lives depend on the active degradosome statistics in the spatial model. So this is something that there's a linkage here between the two. The well-stirred model has longer half-lives on average than the half-lives in the spatial model, as we see here. And they don't really say why that is exactly, but there is also agreement between the well-stirred and the spatial simulations. And the mRNA counts in the spatial model are higher on average, likely due to the difference in transcription rates between the two models. So it's the spatial model also has lower concentrations of nucleotides, and then we talked about that. So yeah, there's a lot of, like, there's agreement in terms of like that those two models are modeling the same thing, but there are differences in what's going on. So you have all sorts of, with protein counts.
Yeah.
Yeah.
The differences are not great.
Yeah, not really, no. And it's, it's, again, I think maybe the assumptions that are made under each condition too are important.
Yeah. Okay.
Yeah. Okay, so yeah, Jesse was here. Well, thanks, Jesse, for attending for the period that you did. So do we have anything else to say about that paper? I was just big picture.
Okay, so basically the idea that diffusion is sufficient to move things around
Yeah.
seems valid, and so the non-spatial model may be adequate.
Right.
Okay, so the only question is, did they actually simulate cell division?
Well, they did. I don't know if they simulated it like under a number of conditions. I think there was an assumption of a cell division. Let's see if they have it in here. Let's see. They talk about the cell cycle here. I don't know if they actually simulated the cell cycle though, the same way they simulated some of this other stuff. At least not in this paper. I don't know. There are a lot of papers on this, so they may put it in another paper. Yeah, I think their focus here was on some of the internal mechanisms of the cell, and what's going on there. As they have this, well, actually, so they have like a cell volume over time, like from 0 to 100 minutes. So this is like a single cycle, I think, where it's like they're going from a cell volume of a single cell to something that's dividing. But this is like one division cycle. So they're just looking at the cell volume over that interval.
Okay. Simulated cell volume is proportional to total number of molecules, basically?
Yeah, I think so. Yeah.
Okay.
Yeah. And then, yeah, again.
Did the model include synthesis of the membrane?
Oh, I don't think it did. Yeah, I don't think they did that. I think it's kind of. So this is the model here.
So how does it divide without its myosin and microtubules?
I'm not sure. I guess that they just kind of.
FTSZ.
What's that?
FTSZ.
Oh, okay. So that's the mechanism.
Presumably they got it.
Yeah.
Yeah, the prokaryotes use FTSZ. They don't do it the way eukaryotes do.
Yeah.
FTSZ FTSZ is actually similar to microtubules.
Okay, yeah. Nice.
Except it can produce a constriction.
Okay, it's a nice cytoskeleton course.
There's a lot of literature on it. Just look up FTSZ.
Yeah, okay, well,
FTSZ for Bradley.
Yeah, wow.
Right. Where's my notes? I was pretty sure it was included. No. Wrong notebook. Oh well, got too many notes. No, that's not it. Oh well, it went somewhere else.
Okay.
Anyway, I'm sure they included it in my course on cell cytoskeleton, so they didn't go into enough depth. It was just one of those small Coursera courses.
Oh.
Yeah.
Anyways, that's okay.
Yeah. Well, I think that was a really good paper for understanding how people are simulating cells and having the parts there in a very simple cell that would describe sort of a function in the cell and what you need there and how those things behave. So, I mean, any larger insights, I know the whole point of this was to look at sort of the origins of life or the origins of a cell. So,
Yeah, but that's not their point.
they're not what they're doing, of course. Yeah, they want to actually know what like the minimal function is. But our purpose is for looking at this, I guess.
Bradley, by the way, has anyone invited Levin to give us a talk? Steve Levin?
Steve Levin?
Yeah, Steve Levin.
Yeah, I guess so. I haven't gotten around to.
Well, Stephen M. Levin, he sent me an email, so I could send you his email.
Okay.
Yeah, I think it might be a good way to see what he regards as the truth of tensegrity.
Yeah.
Okay.
Yes, he's very into it's Stephen Levin 1 at mac.com, mac.com.
Okay. So
Yeah,
he doesn't even have the
Once you
app.
send the there's no problem in sending correspondence to Bradley.
Yeah, that's good.
Yeah, I suggested
Oh, okay.
that he give a talk in DevoWorm.
Yeah.
Yeah, see if he'd be willing. Like he says he's 90 now.
Yeah. His email predates the apple.com domain, some hack.com. Or is that a...
I don't know. Anyway, the emails are about biotensegrity, and he has been following some DevoWorm. He said he dropped in on a session at one point.
Oh yeah, probably. It was a long time ago. Yeah, I remember his work from what was it, the embryo physics course that was they posted about
Okay.
a decade ago or now.
Yeah, it could be easily.
Yeah.
Yeah, I spent a week with him.
Okay, yeah.
Yeah.
Okay, but he says biologic matter is a condensed soft matter and has the properties of a gel, and then therefore conforms to Plateau's law on foams. Okay.
Yeah, some of this needs to sink in.
And I don't know what that is, so I better look
I
at it.
know, that's why we need to get them.
Yeah.
Okay.
Yeah. Okay.
Okay. Well, that's anything else anyone wanted to mention before we go? I think that was a good meeting.
Okay, Jesse came back.
Oh, that's Morgan actually.
Oh, is that
Hello,
Morgan?
Morgan. Yeah.
Right.
All right. Well, thanks for
Okay.
meeting. All right.
Okay, thanks a lot.
Talk to you
Bye.
next week. Bye.
Bye.
Bye.